Hi, I'll wait for another 3 minutes before I start sharing the screen and start with the session. Okay, I'll start with the session. If people join, I'll start accepting them after that. But in the meanwhile, if you guys have any questions, please do let me know. We still have, after this session, we'll have two more sessions, and that will be the end of it. And then you'll have your exams. Let me start sharing screen now. Is my screen visible in full screen mode? Could someone unmute and confirm?
Yes, ma'am, it's visible.
Yeah, okay. Thank you so much. Yeah, so today is the 10th session. Let me just quickly go to the slides. So this is going to be a continuation of bioremediation, specifically to look at different aspects of biodegradation. Uh, just a second. Yeah, the full screen is still visible, right?
Yes, ma'am.
Yeah, okay, thank you. Yes, so biodegradation. So this is a process that is different from the rest of the bioremediation that we have so far seen. We've seen biostimulation process, bioaugmentation, but biodegradation is something that we often hear in terms of biodegradable products, and even in plastics and materials, it's always written like it's biodegradable. So what is this biodegradation? So to simply put, this is an image from Wang et al. They call biodegradation a self-purification process, because this often happens automatically in nature. Nature uses various microbes present in the source of contamination to purify itself. So that's why biodegradation is a self-purification process, and this is done on high molecular weight compounds, long chain compounds like plastics, which are plastic is basically polyethylene. Ethylene is a carbon molecule, and polyethylene is just multiple ethylenes combined together. So that's what plastic is. So it's a higher molecular mass compound and it is fragmented into smaller processes. So biodegradation as a process involves these four steps: fragmentation, abiotic hydrolysis, biotic hydrolysis, basically hydrolysis, assimilation, followed by mineralization. So this fragmentation could happen because of various reasons. It could be because of UV radiation, mechanical forces, microorganisms. Mechanical forces is just wear and tear and stress. UV radiation also includes rain and high temperature, sunlight, and so on, and other all the other climatic conditions which could break these higher molecular weight compounds into smaller fragments. So that's fragmentation. But this could also happen because of microbial activity, microbe which can specifically act on these and cut them up into smaller pieces. They may not be able to degrade them, but like degrade them fully, but they may be able to just fragment them and start the process of biodegradation. This is why the thickness of the plastic or polyethylene is very important. The ones that are really thick won't get sheared or fragmented and won't be degraded. And that is why some of these micron thickness etc are mentioned. And even if it is too thin also, it'll be difficult for any forces to act on them. If they are too thin, they'll just stay there and without being cut up, because it should be of appropriate thickness and appropriate micron sizes, so that they'll be acted upon by these various forces. So that's the process of fragmentation. And then we get microplastic. Microplastic does not just come from these bigger ones, but microplastic itself is present in other present in other sources. There are like these microplastic beads that are there in cosmetic products, usually face washes. If you use face wash, you can see that you have these tiny bead like things in the face wash, which they claim for better cleansing and purification and so on. But those are usually microplastic beads that are used for all the scrubbing purposes and all of that. And those usually end up in waste as microplastic. And this could be high and low molecular weight. The bigger fragments might still be higher molecular weight, but the microplastics might have actually lower molecular mass. And then more enzymatic action and more microbial action is needed after this process, because after they get fragmented, they need to be hydrolyzed. Either abiotic hydrolysis again means because of temperature and other external sources that cause hydrolysis, and biotic hydrolysis is because of enzymatic components, especially that are done by microbes. So after this hydrolysis is basically breaking of hydrogen bonds, hydro plus lysis. And once this happens, they are broken down and shared into more soluble intermediate components. It could be either oligomers, or dimers, or monomers. And after this small shearing also happens, the next step is the assimilation process. So assimilation is where whatever the smaller broken down products are, they are taken as substrate by enzymes, and those substrates are then converted into biomass. And in the process, carbon dioxide and water is released. That's any standard metabolic process. So this substrate should be utilized by the microbes to process them into biomass that is harmless. And mineralization is basically the formation of CO2 and H2O, and this will in turn form other salts and so on. So this process is called as mineralization. So after assimilation, after being taken up by the microorganisms and used as a substrate, they should be broken down to microbial biomass, which is harmless to the environment. So this process is happens naturally in any given environment, and that is biodegradation. But not biodegradation may not always happen organically. We may have to push it by adding some of these microbes externally, and that is what we call as bioremediation. So we mentioned multiple factors here that are responsible for bioremediation. So along with the exact bacteria or microbe that is required, there are also other essential factors which support the microbial growth, like pH of whatever that contaminating source is. It could be wastewater or soil, it could be any of these sources, but all of those need to be at a pH that is convenient for the microbes to grow on, and also the buffering capacity of the minerals, whatever phosphates or sulfates or whatever sources, it could be copper, manganese, so on and so forth. All of these minerals that are present in whatever that source should be able to buffer in such a way that the pH and everything is appropriate for the microbe to grow. And most important is moisture and temperature, because this is what determines exactly whether a microbe will grow in that particular temperature or not. um yeah, just a second. All right. Yes. So the most important thing, or whatever that are the restrictions for a process of bioremediation, is the toxic impacts that the byproducts will have. Most importantly, before even doing bioremediation, we have to check if we use a particular microbe, whatever product it releases after the substrate utilization is has any impact on the environment, whether that itself is toxic. And then whether it interacts with, so here they have given an example of petroleum, it could be any waste product, and the interaction between the microbe and that substrate is most important. Environmental constraints, metabolic constraints. Metabolic constraint again means that it might be able to take in that substrate, but it might not have the essential enzymes that can actually break down the substrate. It might probably need some additional help of other microbes to break down certain intermediate products which can't be broken down by the original microbe itself. So those are certain metabolic constraints. But this whole process is time consuming. We can't just expect the microbe to just take in the substrate and convert it in within a couple of hours. So bioremediation is a time and a time consuming, and the most important rate limiting step is how the metabolism of the microbe itself works. So these are some of the factors that we need to take into account before we actually choose the microbe for a particular bioremediation process. So the like I mentioned in the previous slide also, here this is an example of a metabolic pathway, how a metabolic pathway progresses. So initially, let's say this is the first or the main pathway. There is one substrate, and it gets converted to another substrate by enzyme 1. Similarly, from B to C, C to D, and D to E, totally four different enzymes are required to convert the substrate A to product E. So this is the initial reactant, so that's the substrate. In case if the microbe that we choose it does not have enzyme 2, the process will stop at B. It will not progress to C to D, and then finally to E. E is our desired product. That is probably the product which is less harmful to the environment, and we want to make sure this is what we get at the end. In that process we have to choose a microbe that will go through these full processes with all the four enzymes present in it. But another important thing to note is that this pathway could also branch depending on the conditions at this point. It might go to D or it might go to F depending on the external conditions. It could be pH, it could be temperature, it could be even the abundance of microbial biomass at that stage, because even the microbial biomass amount at the end also determines how much this process gets shifted towards a particular pathway or another pathway. So all of this is important, and this is why we learned those several techniques through which we can actually understand all of this metabolic pathway through genomic sequencing, transcriptomic sequencing, proteomic sequencing, and metabolic sequencing, we'll be able to get an idea of how the microbe is able to process the initial reactants from the substrate A and reach until the final product of E. These relevant enzymes are needed for degradation of organic compounds. In case of biodegradation, we would want organic compounds like petroleum products to be eliminated, or oil to be eliminated from contaminating water. Then multiple enzymes will be needed for continuing the process. This is an example of how multiple enzymes are needed. We saw this even in one of the previous sessions. This is the proposed biodegradation pathway of benzene by bacteria Pseudomonas. Pseudomonas, as we know, is the superbug, and it degrades.
I'm sorry
Yes, yes.
to interrupt, like, because of my network problem. Can you please slow down and explain this again?
Uh, okay, I like a
I'm
couple of
sorry.
slides or from this slide.
Like, bioremediation is time consuming, from the metabolic pathway.
All right, sure, sure. Yeah, I'll go back to that. Yes. Yeah, this slide
Thank
is
you.
still in full screen, right? You can see the
Yeah,
slides,
yeah,
right?
it's
Yeah,
full
okay,
screen.
yeah.
Yeah.
Yeah, so this is where you might have lost me. So I was saying that bioremediation is time consuming, and the most important thing is that the microbe that we are choosing should have the relevant enzymes for degradation of the organic compound of interest. It could be any organic compound like petroleum products or oil components, or any other metal contaminating the wastewater or soil. But the microbe that we are choosing should have all the relevant enzymes that are required for degrading that specific organic compound until the final product, which is harmless to the environment, could be reached. If we choose a microbe that has one enzyme but does not have the enzyme 2, then the pathway will stop at this place. In that case, we should have another microbe that can take over from here, which is what we'll see in the next slide for the bioremediation of benzene. The most important thing that we take away here is that we should choose the conditions such as pH, temperature. Along with that, we should also choose the microbe in such a way that we achieve the final end products. So this is an example of how multiple microbes are required and multiple enzymes are required for the biodegradation process. Here, the substance that we are converting is benzene, or any benzene containing molecule. So let's say this is a waste from one of the industrial effluents, and it is in water, and we want to degrade this benzene in the water. Then one can use Pseudomonas is called as the superbug, Pseudomonas putida, and different strains of Pseudomonas are used for degradation of oil spills and so on. Here in this case as well, Pseudomonas can degrade benzene to phenol, it can be the same strain or a different strain which can further break it down to catechol or hydroquinone. But Pseudomonas activity stops here because it does not have further enzymes to process catechol or hydroquinone. So from there, it will be taken over by acetogenic bacteria, which will further process it into benzoquinone and then malic acid. And from where methanogenic bacteria can take over and convert it into oxalic acid and carbon dioxide and water. So in most of the metabolic pathways where we want to achieve a biomass that's safe, we have to degrade it up to the point we reach carbon dioxide and water. That is when we know it has been degraded to the maximum extent, and it has been broken down into the lowest possible organic compound that is not harmful to the environment. So this is a multi-enzyme process for degradation of benzene. So therefore, choosing microbe or set of microbes that can degrade at various stages is the most important thing, along with the other abiotic factors and so on. And another important thing that we also have to keep in account is the CNP ratio, carbon nitrogen phosphate ratio, in the soil or water or wherever the source that needs to be converted is. This CNP ratio usually comes from the substrate amount itself. And if this ratio is not ideal for the microorganisms to grow, especially for the microorganisms to grow in places like aquifers, where we want to grow them at a particular condition, then the CNP ratio should be 30 is to 50 is to 1. And this CNP is what will determine whether the growth is maintained, whether they are growing or whether they are dying. And this CNP uptake and release can be continuously monitored so that the source can be enriched with the nutrients required for the microorganisms to thrive in that process. This just gives us an overview of the CNP is turned over during the microbial growth and death and maintenance of the growth itself. And We spoke about enzymes, the different enzymes that are required. And most common and the largest class of enzymes that are used by microorganisms are oxidoreductases, and hence these are also the enzymes that play a major role in biodegradation as well. They are a large family of enzymes. They have multiple functions. They can be hydroxylases, oxidases, dehydrogenases, reductases, peroxidases, and oxygenases. These are all just different things that is got to do with oxidation and reduction reactions. It is either one or combination of one or two of the oxidation and combination in various ways. And How many units of oxygen is added or how many units of hydrogen is removed and so on will determine what class of enzymes are required for that particular process. But the most important thing to note is that oxidoreductases are the largest class of enzymes that we'll often encounter in any biodegradation process. One such example is reduction of polychlorinated hydrocarbons. Polychlorinated hydrocarbons are often spoken about like CFC effluents from AC, refrigerator, those coolants and all, and so on and so forth. So to capture this and reduce it into something that is not harmful to the environment, that will not cause air pollution, is the challenge. It is the biodegradation that needs to be carried out. And here as well we see that most of the enzymes that are involved in the reduction or conversion of polychlorinated hydrocarbon is are reductases, and reduction is the reaction that happens for biodegradation of polychlorinated hydrocarbons. Another example where reductases come into play is conversion of chromium. Hexavalent chromium is a very harmful pollutant. We can see that these are all the different sources from where hexavalent chromium is a metal, a heavy metal, that it comes to the environment either through direct industrial inputs. Volatile organic compounds can contain chromium molecules, and industries could just be processing something, especially like leather industries and all. They might be releasing this directly into the environment. It could be from natural oxidative processes in the soil. It could be from agricultural activity and so on. So these are all different sources from where chromium would enter into the environment. And the major health effects of chromium include skin diseases, skin cancers, lung cancers, perforation of nasal cavities, bronchitis, asthma, so on and so forth. Uh, so yeah. Yeah, here as well we can see that the person's x-ray is shown, where they have chromium based perforation that has happened, and also lung cancer that has happened because of extreme exposure to the chromium 6 compound. This chromium can be biodegraded by microorganisms, again through reduction, and these are all the different processes through which the chromium goes through in microbial biomass, and how it is broken down in the chamber with the help of microorganism and processed into compounds that are less harmful for the environment. And these are all the different processes that happen inside the microbe: ion exchange, adsorption. The chromium is adsorbed into the surface of the microorganism. Chelation, where the metal ion is chelated for complexation and then further precipitation. So these are all different chemical processes that happen for the degradation of chromium inside the microbial cell wall. So here as well, we can see that oxidation reduction is the main process, and there are several enzymes that are required in each of these processes, where chromium is taken in from these different sources, and they are converted into various products, various intermediate products, and so on. So here as well, the take home is that oxidoreductases play a major role in conversion of hexavalent chromium, and a reducing microbe is what is required, or a reducing metal eating microbe or a metal converting microbe is what is required in this process as well. And when as we are talking about metals, metal eating microbes and so on, I'd like to bring your attention to one of the metals that is highly contaminating. Yeah, just a second. Yes. Another metal that is often found in water or any of these contaminant sources is copper. Copper actually is one of the metals that helps in microbial activity to a certain extent. Some microbes even require copper as a cofactor for many of their enzymatic reactions to fasten the process. But copper works both ways. We all know that copper is really good for health, and even keeping water in copper vessel is suggested because it's a cofactor for several reactions, and it is also known to be an anti microbial, it known to have antimicrobial properties, antifungal properties, and so on. That is at a higher concentration. As the concentration of the copper starts increasing, it stops being just a cofactor for the microbe, and it starts becoming toxic to the microorganism itself. Why is this important to consider? Because if there are copper in the source in which we are trying to do biodegradation or bioremediation, and if it has copper, depending on the concentration, it is going to either help the microbe of our interest, or it is going to cease the activity of the microbe itself. Therefore, it is essential to look at the concentration before we actually use that source, or use that particular microbe for that particular source. And as we are looking at how metals have a problem with microbes, another thing to look at is how the microbes themselves have come up with mechanisms that they can use to get rid of metals. And these processes, if we understand these processes in detail, it will help us really when we are trying to contain a metal contaminated zone and so on. We can take some of these factors into consideration and use these factors while doing bioremediation at a source. Metallothionein is a compound that is found in microbes that help in containing the metal toxicity at lower concentrations. It is a low molecular weight sulfhydryl rich protein. Metallothionein is a protein that is produced by microbes, and it helps in combating metal toxicity in lower concentrations. And it is also found in eukaryotic cells, not just microorganisms. It's found in higher organisms as well. It performs this free radical scavenging activity. it also acts as a zinc donor for zinc finger transcription factors. It takes up the zinc and repurposes it for this action. It also has an activity on various other processes that happen inside the cell. It is mainly for maintaining the proper metabolism inside the body, and hence plays a role in various processes like survival, growth, drug resistant, and differentiation, and so on. So this is a process how the metallothionein works, how it bioaccumulation of lead by metallothioneins. This is a process how metallothioneins help in bioaccumulation of lead, thereby preventing lead toxicity. So we can also use this in places where it is contaminated with lead, like a soil contaminated with lead, or water that is contaminated with lead, and we will be able to contain the lead contamination in that particular place using metallothioneins. So the metal ions are basically absorbed into the surface of the cell, and from here it leads to intake of those components, bioabsorption of lead on extracellular polymeric substances. So these are the extracellular polymeric substances, EPS, and it takes up that, and it takes the lead inside the cell, and from here it is processed inside the cell. It is broken down by enzymes, and basically metallothionein performs this, and the broken down products are later on precipitated, and it can be further used for some other useful process, or it can just be excreted from the microorganism cell wall. So therefore metallothionein is a very important protein that can help in containing metal toxicity. Yeah, so this is mostly what we have for this session. We'll be continuing with a little bit of biodegradation in the coming sessions, but today's session, it's mostly about understanding these different factors that are take that should be taken into account when we are trying to understand how we can control toxicity of various contaminants, and most particularly metal based toxicity and metal contamination. We will look further into this in one more session, but yeah, so this is mostly what we should know or keep in account when we are trying to consider degradation of metals by microbes. So yes, if
This is the first time I'm attending this session.
okay.
So all these contents will be available in the NPTEL videos, or these are the extra contents.
So the NPTEL videos are by the professor. Those lecture content is different, but it's so whatever these sessions are mostly like a summary of what is there in that whole week session. That whole week session will have like 4 hours videos and so on. So this 1 hour is just mainly to clarify doubts if you have any doubts in the assignments, and these are like mostly like the solutions for the problems that will be generally asked in the assignments and also in the final exams. So this is to get an overall idea of what is there in this week's content. But this PPT that I'm using, this is also available in the NPTEL portal itself, in your page where you have the course content. If you go towards the bottom, there will be an
Yeah, in the
option
last
problem.
class you showed us.
Yes, yes, yeah. Problem solving session, and in that you have a link to the Excel file where you'll have links for the presentation, and this video also is recorded and it will be uploaded. In case you have any network issues, you can always check this video. It will be there on YouTube. You can just check that, or if you want the video, you can also download it. It is available on as an external file as well in the same link. So you can get all of this information from the Excel sheet itself.
Okay, thank you.
If you have any further questions, or if you were solving the assignments and if you are having any doubts from the assignment questions, you can ask them. um, we can discuss those as well.
So the role of metallothionein is also like bioremediation.
Yes, so metallothionein is a natural compound that is there in microbes, in many microbes. This is there for controlling metal toxicity for themselves. Microbes grow in various conditions in the environment. They can't always expect any external sources to control the metal concentration for them. So they have this property where they have this protein called metallothionein. They also do other forms for eliminating metal ions from their sources. They do it by bioassimilation, they do it by precipitation. They have certain enzymes that can precipitate the metal ions. They also have ways of breaking down metal ions. Metallothionein is one such compound that helps in processing of the metal ions by taking up metal ions and then releasing the precipitate. So this is a compound that can be utilized in the bioremediation process. If we are planning to use this, we can choose a microbe that has metallothionein content, and hence it can contain the metal toxicity. And this can also be used in cases where our target product or target substrate is different, but there is a lot of metal contamination as well. We can enrich metallothionein protein in the particular microbe so that the metal toxicity is taken care of, especially in cases like copper, where we need copper for the microbe to grow. But if the copper concentration goes very high, it will start becoming toxic for the cell itself. In such cases, we can use metallothionein. We can enrich the microbe with metallothionein so that the copper toxicity is controlled.
Thank you.
All right. Are there more questions, doubts from any of the assignments? So I've again seen mails from people mentioning about assignment solutions not having the right answer and all that. So please use the lecture slides. They have the right answers. They are the actual solutions. So if you are able to find the solution in the lecture slide itself, please do use that. The NPTEL team will definitely release the right solution if there are any mistakes. But for your exam preparation, please do use the actual course content lecture slides.
But how have you provided your mail ID? I didn't receive it.
Uh, I have sent it in the previous sessions, but yeah, I'll just send it again. You can always just mail in the discussion forum. I also receive those mails. You can always mail in the discussion forums, and I can respond back if you have any queries. Even during the course of the week, if you have any queries, please do send a mail in the discussion forum or to my mail ID as well. I can definitely try resolving if you have any doubts. Uh, okay, if there are no more questions, we still have two more sessions to go. It will be next Tuesday and the Tuesday after that. And if you guys have any questions throughout the session, um, please do keep them ready and ask them during the sessions. This doesn't have to be from that particular session. It could be throughout the course content. If there's any questions, please do keep them ready, and we can discuss them during the session itself. All right. Thanks a lot for attending. Thank you guys. Bye bye.
Should I have to like view all the videos from the beginning, or still now I have not started?
Okay. So
So I can start in between also
the videos as in the lecture videos from the course content, or these videos of the problem solving sessions.
from the course content.
Okay. So if you're actually taking the exam, if you have registered for the exam, you should go over all the content
Yeah.
from the beginning. Uh, because the questions will be asked from all the sessions throughout the 12-week lecture content. So please do go over all the watch the videos from the beginning. And these problem solving sessions are just a gist of the overall idea of what is there in each of the week. You can use this to get that overall idea, but to answer specific questions for the exam, I would strongly recommend you to go back and read, like, go over all the lecture content from the NPTEL website, and also try
So
solving
the
these.
lecture notes are also enough, right?
Sorry.
So it's enough that we go through only the lecture notes, right?
The lecture notes and the videos as well. If you are not understanding a concept, it's better you go over the videos. I think that's, I
Okay.
mean, if you look at the PPT, some of you might miss on some of the information that is given during the video recording session. So it's better to go over all the videos. So try to do that as much as possible.
Yeah, thank you.
And try to solve all the assignments as well. So many of these problem solving sessions are from the assignments from the previous years, previous course run. So this will give you an idea of how the questions will be. So please to try to solve all the assignments from each of the week content, so that you get a better idea about how the final exam questions will be. Okay, all right. I hope your doubts are cleared. Please do write to me or get back to me in case you have any more queries. Thanks a lot for attending today's session. Thank you.